Tejas Shah. He is a consultant at X-Claris Lab, Ahmedabad, and fellow of Department of Animal Biotechnology, AU Anand. He has completed his BSc in Genetics from NVPAS College, SP University, and MSc in Biochemistry from Institute of Science, Nirma University, Ahmedabad. He has 10 publications in reputed journals. He has qualified GATE 2009 with 95.5% score. So I welcome Mr. Tejas to please come and continue lecture.
Thank you, Reena. Good morning to everyone. Today I am going to talk on the Human Microbiome project. So this project is the second largest initiative by the NIH after the Human Genome Project. So I am trying to summarize this project to some extent level. So we all know that our body is marvelously intricate machine, which parts includes the trillions of microbial cells that colonize our epithelial surfaces like gut and the mouth. So it has been recognized that these microbes do more than just reside on the gut, reside on the tissues. They play key roles in the human physiology and the organ function. There are 100 times more genes in our microbiome compared to our human genome, and these microbial genes code for proteins that impact the diverse processes such as the digestion, immunity and the development. So the attempts to understand the human associated microbial communities were given the boost by the Human Microbiome Project initiatives at USA, Canada and the Europe. So this is the time of the unprecedented discovery, and although still it's young, the field is sufficiently advanced. So today I am trying to summarize the Human Microbiome Project and its progress. So now we all know that what is the microbiome. So it is a full complement of the microbial species and their genes and genomes that inhabit the host body. So NIH Human Microbiome Project is completely community resource project. I repeat, it's completely community resource project, and why it is completely community resource project that I will discuss in the later slides, which is designed to promote the study of the complex microbial communities involved in the human health and disease. It sometimes seems that the research on the human microbiome blossomed overnight, but however, the conceptual and the technological foundations for the study of the human microbiome began to emerge before the 90s and can be found within the many disciplines. It was microbial ecologists recognized early that the most microbes in the nature are not culturable. So they developed the alternate approaches, and the early and the broadly adopted approach is the 16S rRNA, that is the taxonomic marker gene used for the such kind of the environmental microbiome community analysis. So with the growth of these non-culture based molecular techniques in 80s and 90s, some medical microbiologists turned these tools to the human body, and they found far greater diversity than they expected, even in the oral cavity. So it has been in the infectious field, it has been recognized, growing evidence that the many diseases could not satisfy the cause postulate. That is this number of diseases, infectious diseases are caused by the many microorganisms. But that's why the polymicrobial disease term was coined, that the number of many diseases are caused by the many microbial factors. So for example, AIDS related infections, opportunistic infections, multiple sclerosis, conjunctivitis, enterocolitis. So there are these polymicrobial diseases are there. So now we look at the history of the NIH. So it was established in the 1887 for the testing of the infectious disease samples, that is purely microbiological work. And then in mid-20th century, this lab helps to crack the genetic code and leads the Human Genome Project. Now it's 21st century, it coming back to the microbiology, and it studied the ecological approach to our fellow travelers, nothing but our conventional microbiota. So the Nobel Prize winner and the NIH grantee Liederberg proposed the term microbiome. So he defined the microbiome as an ecological community of the commensal, symbiotic and pathogenic microorganisms that literally share our body space and have been all but ignored as a determinants of the health and disease. He also said that we should think of each host and its parasite as a superorganism. The first publication based on the analysis of the genes of the community of the human microbes, that is published in the Science in 2006, demonstrate the feasibility of this initiative and this kind of the study. So the time these people have realized that this is the right time to undertake such a comprehensive study, and in 2005 Indra organized the meeting, the scientists from the USA, Europe and the Asia, and they discussed on how to approach such a study. This two-day meeting covered a broad range of the topics, including they all recommended that we have had to sequencing all the bacteria, and what would be the impact of the human microbiome on the study of the health, and what would be the possible structure of the human digestive tract microbiome program. The recommendations coming from this meeting, that is the formation of the International Human Microbiome Consortium, and they all agreed to that the data, we have to release the data as rapid as possible, then share the data standards and develop the reference data sets as early as possible. So this discussion led to the formation of the European Commission's call. During this period of time, National Academy of Science published a report on the metagenomics, which highlighted this new field and informed the scientists that what is the potential of this advanced area. So during this period of time, NIH also invited the community comments, and they have also identified what would be the specific needs of this project, and they have identified a number of needs that require to first follow. That is a reference microbial genome sequence catalog, animal models for the microbiome studies. We need to study the benchmarks analysis of the 16S rRNA and the microbiome metagenome sequencing, the computational tools to analyze this new kind of the data, and the ethical aspects of the human microbiome research. The pilot projects to develop the protocols on the sequencing of these human metagenomic microbiome samples, that is begun in the mid-2007 by the National Institute of Human Genomic Research. So in 2007, that is NIH Common Funded Support Human Microbiome Project was officially launched. So this project is with the intent to produce a number of major community resources, a reference microbial genome sequences, a large cohort study to survey the microbiome across the human body in the healthy individuals, a suite of demonstration projects to examine the correlation of the changes in the microbiome with the disease, and the computational tools to analyze these kind of the data. And this project lead to the international effort, and number of other countries are also involved in the this kind of this study. After the launching of this HMP project, the MetaHit project was launched by the European country, that is Metagenomics of Human Intestinal Tract, and all these countries are specific to the body sites. So these are the three major initiatives of this human microbiome project. They have to sequence the microbial sequences that inhabited the human body. Then they have to sequence 16S rRNA genes from these 300 subjects across the 18 body subsites and the shotgun sequences of these 300 subjects across the 18 body subsites. So, now come to the guiding principles and the structure. So, as I have already mentioned previously that this is completely envisioned as a community resource program, and they have defined this community resource program is a project that specifically devised and implemented to create a set of data, reagents or other materials whose primary utility will be as a resource for the broad scientific community. It has been recognized that this metagenomic and the metadata that is collected by this HMP project, these are the unique research resources, and in order to establish and serve as a community resource project, they have included the rapid data release. They have to follow the guiding principle that include the rapid data release into the public databases, and they follow the guiding principles that created for the Human Genome Project and all of the large scale genome project that has been done at the NIH.
Okay, Tejas, can I ask you a question?
Yeah.
Where do this Human Genome Project and the Microbiome Project actually meet?
When they have launched the draft genome sequence of the human genome, then they have made a sentence that we have to sequence our second genome.
So they are saying that second genome is microbiome, right?
Yeah.
But in microbiome, what did they consider? I mean, because in our system, as a human body, we have a gut microbiota. We are having different organs on our skin, in cavities, different cavities. So in this microbiome project, did they consider all these points?
Yeah. They have considered 18 body subsets that I have discussed with you in a later slides. So they have also established collaborative activities. For that, they have established the HMP Research Network Consortium to analyze this large scale data, and the product of this is ultimately to the benefit to the community resources. So these data usage agreements are posted on the DAC website. From the healthy cohort study, it was recognized that the whole genome sequence of the human whole genome shotgun sequencing of these metagenomic samples would capture the human genome sequence also. So they have decided that these sequences would not be made publicly available, and when we are going to this in a NCBI, then we have to remove these sequences from the metagenome samples. So for that, they have adopted the NCBI DBGap database, that is the database of the genotypes and phenotypes for the HMP clinical metadata and sequence data. It has a two level of access, open and control, in order to regulate the sequence and the health information of these volunteers. This open access contains the publicly accessible databases, whereas control access requires the permission from the NIH Data Access Committee to access this data, clinical data as well as the human data from the metagenome sequences. So these targeted 16S rRNA data are present in the open access database of DBGap. As I have mentioned previously, that the other international agencies are more focused on the specific body sites. So they have decided to survey, these HMP people have decided to survey the microbiomes of multiple body sites in the healthy adults and to generate the baseline data and develop the catalog of the microbial genome sequences of microbiome reference strains and evaluate the association of these microbial communities with a specific disease. For that, they have also created the data analysis and coordination center to manage the data from the sequencing activities. They have processed the sequence data to the consortium, agreed on the standards for the further analysis. Whatever the pipeline they have decided, then the same pipeline they have used for all the analysis of the samples. They also coordinate the data analysis activities in the consortium, and they also serve as a portal for the scientific community to access the dataset, tools, and the other resources. So there are the three resources from that we can get the information of the HMP program. The first is the NIH Common Fund website. From that we can get the overview of the main initiatives of this HMP program. Second one is the NCBI Bioproject site, where what kind of the data they have produced. They are listed in this NCBI Bioproject website. Based on that, 4 projects are linked to the NCBI Bioproject under the HMP umbrella based on the data they have produced. The first data they have produced, that is the 16S rRNA gene data, whole genome short-term metagenomes dataset, reference strain microbial genome sequence datasets, and dataset produced in the individual demonstration project activities. So, in order to establish the scientific approaches and protocols for the sequence of this large number of samples, these human microbiome samples, they have 4 large sequencing centers: the Broad Institute, Baylor College of Medicine, Washington University in St. Louis, and JCBI Institute. So these 4 institutes are responsible for developing the protocols for the DNA extractions. They are sequence the microbiome samples as well as the reference strains microbial samples. They analyze the microbiome samples and contribute to the analysis of the healthy subject microbiome data. They also support the sequencing activities in the server of these demonstration projects. It was recognized that the large-scale analysis of these new and complex data sets, particularly the healthy microbiome data sets, would add value to the resources emerging from the program, and this would require the number of large group of scientists, and for that they have made the data analysis working group, and this group consisted of the combination of HMP grantees, experts in the scientific community which is specifically expertise in the metagenomic analysis, and those all who joined the research network consortium. And during these 2 years of active data processing, this group continuously in contact with each other. They are grouply meet weekly on the conference call. They held the biannual research network consortium meeting, where virtual meeting, that is the 1-day online meeting. In that they have discussed that microbiome data analysis to the experts in the microbiology and the body size specific clinicians to interpret those data. They have also exchanged the computational tools, analysis and draft manuscript through consortium agreed electronic resources. So here at the time they have 200 members of the 20 work group. They are tackling the specific task, and this group work also together to achieve a major goal. For example, the strain this strain working group works with the annotation and the finishing working groups to coordinate the selection, sequencing and annotation of the reference strains of the project. So the HMP sought to create the public reference dataset of the microbial genome sequences from the whatever the body sites they have sampled, and the goal was to create the catalog of the genome sequence from 3000 bacterial strains and as many as viral phage and the eukaryotic microbial strains as possible. But their priority is only the bacterial strains only. And this data are intended to provide the reference for the interpretation of the 16S rRNA sequences, and this data also serve as a scaffolding for the assemblies of the metagenome sequences. An extension of these public resources, this culture of the sequence strains deposited to the HMP repository with NIAID-PE, that is Biodefense and Emerging Infectious Research Repository. So all the strains are available at NIAID-PE. So approximately 100 of them are in the high demand, and this will be in the self-ready state and will be available immediately to the scientific community. And another are in the archive state. When the requests are available to the VIA, they have prepared and make it available. So they have also prepared the guidelines how to select the strains. So they have this, based on these 5 guidelines, they have select their strains, which strains we have to go for sequencing. So these are the criteria: its phylogeny and the uniqueness, its established clinical significance, its abundance or dominance in the body site, whether these identical species were found in the different body site or not, and whether there was an opportunity to explore the pan-genomes.
What are pan-genomes?
Pan-genomes are the core genomes of the microbial species, okay, all the microbial species, and the one or more other genes that are specific for the other strains. So there are the core set of the genes of that microbial species. So from these microbial strains they have sequenced. Majority of them are from the gastrointestinal tract, then followed by the oral, urogenital and the skin. They have completed and almost finished 34 microbial strains, and majority of them are in the high quality draft sequences. And total they have up to 2644 are in the pipeline. And the first, that is the catalog of the reference genomes, are published in the Science 2010. That is the 178 microbial reference sequences are published. And from that they have got around 5 lakh genes, and 30,000 of them are the novel. That is the Human Microbiome Jumpstart reference strain consortium. It was said that the 60% of the human associated microbes are in a culture and they are available for the sequencing. So the technology development initiative aimed at isolating the uncultivated microbiome was created. So this program included the support for the innovative cultivation techniques to isolate the new strains from the body sites, and the application of the sequential cell genomics methodologies to reach this project goal.
So they have already started culturing them.
Yeah. So, in order to guide this effort, the strains working group has conducted analysis of the healthy microbe 16S data to develop the priority list of the top 100 most desirable bacterial sequences to target for the sequencing. And this approach was used to identify the new or novel taxa that has not yet been sequenced, was to select the 16S sequences for all body sites that had a less than 90% identity to already sequenced strains, and that were found in at least 30% of all samples from the particular body site. So from these data they have identified the body sites and that contain most of the strains that had not yet been sequenced, and this, after that analysis, this resulted into the more than 100 targeted strains, and 73 of them are located in the oral cavity and 30 are in the gut. And particularly these data are being used to guide the technology development team to decide on sorting efforts and their searches for the novel strains. So now come to the healthy adult cause study of the multiple microbiomes. So there are the main differences in the terms they have used to describe the body site, but there are main 5 major body sites, that is oral, skin, airways, gut and the vagina. And within this body site there is the subsites. This oral has the highest number of subsites, that is the 9. So in case of male there is only 15 subsites, but in case of female there is 18 body subsites. So from that they have all the samples they have collected and sequenced. So this study is broken down into three components: clinical phase, that is the sampling phase, sequencing phase, and the data analysis phase. So it is very difficult to identify the healthy individual. So they have recruit the experts in the clinical research and ethical issues, advised on the inclusion and the exclusion criteria for the healthy individual, and two consent forms are also developed for their study. So these criteria are based on these parameters, that the individual has a combination of, they have collected the combination of health history, particularly systematic disorder, the hypertension, cancer, autoimmune disorders. So, and they have also not recruit the individual with that use the antibiotics, probiotics, or the immunomodulars. They continuously use that Modimas index, and physical examinations such as presence of skin lesions and oral and dental health states, and women are required to have a history of regular menstrual cycles. And these subjects are informed that their samples and data are coded to anonymize their study participants, and these are stored at the control access database, and this regulated by the NIH Data Access Committee. We have to take the permission from the NIH Data Access Committee to use this human data. These participants are also consented to allow the researchers to use their human sequence data. But on the basis of that, their identity will not be disclosed to the researchers or the scientific community. So they have sampled all the body sites directly except for the digestive tract. In that, this tool serves as a proxy for all distal gut regions. They have collected the saliva from each subject at each visit. Blood and serum are also collected from each subject at the first visit, and the 1 aliquot of this blood is used for the DNA extraction for the future whole genome sequencing. They have also harvested the lymphocytes and stored at -80 degrees Celsius for the future preparation of the cell lines, and this would add the additional community resources and stored at NHGRI sample repository for human genetic research. Two main sequencing centers, that is BCM and W in St. Louis, only extracted the DNA from the body site samples using the same commercial kit and the standard operating procedure, and distributed this DNA to all the sequencing centers.
Okay, Tejas, which institute in India is involved in this?
No.
We don't have any.
We don't have any.
No.
So what was the age group included in the study?
There are no. In this study, they have recruited 300 adult volunteers that are selected from the. They have screened around 550 individuals. These are approximately 20% of them are self-identified as a racial minority, and 11% of them are Hispanic. So this total pool of the volunteers are split in between two clinical sites, that is in Houston and St. Louis. Houston is BCM is located in Houston, and in the Washington University is located in St. Louis. So these two clinical sites as well as two sequencing centers, and these volunteers are self-identified as they are generally meat eaters, and they have had breastfed at infancy. They have an age group of around 18 to 40 age. They have a BMI range is 19 to 34, and that is the pressure are also normal. That is around 70 to 120. They are generally majority of them did not smoke. So majority of them are students. If you look at the occupation students, that is 150, around half of them are the students. So the enrollment and the sampling of the volunteers commenced in the near about December 2008, and it's completed in October 2010. And of the 300 these study participants, they have sampled from that 270 were sampled twice and 100 of them sampled third time. Okay, and between this first and the third visit, it around 10 months. So, and the deposition of their metadata are in the DB was completed in February 2011, that is after the 4 months of the complete sampling procedure. So, as a part of the pilot projects for this initiative, the sequencing centers took a series of benchmarking studies to determine the appropriate protocols for the sequencing and to approach the consistency of the results across the sequencing facility. For that, they have developed the mock communities of the 22 bacterial species assemblage as a test to evaluate the DNA extraction procedure, the primer selection strategy for the library construction, and the sequencing protocol. And based on this mock community data, they have decided that the primers for the variable region V3, V5 they are used for the targeted 16S sequencing for all the samples. And when the requirement is there, then the V1, V2, V1, V3 and other regions are targeted to amplify the specific bacterial groups that do not amplify well with the V3, V5 regions. So approximately 12,000 samples they have uniquely collected from the 300 subjects. And these samples include the 16S to sequence the 16S rRNA or the metagenome of the microbiota, as well as the urine, blood and the saliva samples are also there. From these 12,000 samples, unique primary samples, they have used 11,000 for the nucleic acid extraction. And this majority of them samples are analyzed by the 16S targeted sequencing by the 454, and fraction of samples that are analyzed by metagenomic short-chain sequencing by using both the 454 and ILMINA. So these targeted 16S sequences and WGS sequences are deposited at the NCBI. This, as I previously mentioned, that 16S sequences are deposited in the open access portion of the DBGAP, and these metagenomic sequences and clinical metadata are deposited in the control access portion of the DBGAP. So these are the number of shotgun samples they have sequenced per body size. The majority of them are in the stool samples, and followed by the tongue dorsum, supragingival plaque. So now the challenge is here. In preparation for the data analysis phase, a group of scientists from the microbiome community, the sequencing center and the data analysis and coordination center, as well as the NIH staff were brought together to form the HMP data analysis working group. They formed it. So as the data, they are continuously producing this data. So this data analysis working group declared the data freeze on the May 1, 2010 on the 16S rRNA sequences. So up to this freezing stage, they have sequenced more than 5,000 specimens across the 18 body subsites, and the 242 subjects they have sequenced. On July 1, 2010, they have a data freeze on the WGS metagenome sequencing, and up to this stage they have sequenced approx 700 specimens across the 16 body subsites of the 5 major body sites.
They are so fast.
Yeah. So this data freeze is because in order to define the common and master data set for the follow-on global analysis activities to be undertaken by the research consortium. So this is the paper published in 2012 based on this data. So they have sequenced near about more than 5,000 for the 16S samples and near about 681 samples in the WGS samples. From that they have around 3.5 TB data for the WGS after removing the human sequences, and they have around 19 from the V1, V3 region and 26.3 million from the V3, V5 regions. So that is around 50 to 60 million reads from this 16S rRNA reads are they have sequenced.
Paper title. Paper title.
That I will give you next time. So for each major body site, the typical sequence generated from the sample range from the 10.8 to 12.8 GB, and the average is the 12 GB. And these data sets are included the total, they have a sequence around 8 TB, but after removing of these human sequences, they have come across around 3.5 TB for the microbial, unique microbial sequences. And up to this date, they have sequenced around approximately 74 million 16S rRNA reads. As I mentioned that the WGS sequence contains the human sequences also. So these are based on the human tissue they have collected during the microbiome sampling. So in case of this tool, that is around, they have got around 98% of the unique microbial sequences. But in case of this vagina and the skin, they have got around 10 to 25% only microbial sequences. So first they have removed those human genome sequences, and then they have deposited this data to the open access portion. So this is the pipeline available on the HMP website. So they have done the 16S rRNA analysis using this RDP that we have already learned in this workshop, through and the KIME. They have used these 3 different pipelines. And for WGS sequence data, they have first processed this WGS sequences by using quality filtering criteria, and these 454 rates are used for the 454 plus Illumina rates are used for hybrid assemblies using the Nimbler, and these only Illumina rates are used for the assembly using this soft de novo. So these assemblies are used for 2 kinds of the metagenome analysis. As these assemblies are used for the analysis of the comparison against the reference genome sequences or to determine the microbiome community composition, and the hybrid assemblies are used for the reconstruction of the metabolic modules and the pathways informed from the whole genome sequence data. So now come to the some results part. So as they have got more number of new microbial taxa, there is no any increase of the new gene clusters. So here they have a ratio of the gene cluster, unique genes with a ratio to the unique genes to the unique OTUs, and these are stable to for this tool and buccal and buccal and the anterior nets that are the stabilized, whereas in case of the supragingival plaque, with increase in the unique OTU, there is no any increase in the unique genes. Based on this alpha diversity, this oral and these stool samples have the highest microbial composition compared to the vagina and the skin. But when these samples are compared to the other samples of the same habitat, that is the beta diversity, in that the oral samples have a lower, more or less beta diversity, and it suggests that this microbiome is fully rich with metabolic functions, and this microbiome is shared with the other members of these subjects. But in case of the vagina, it has the lowest alpha diversity as well as lowest beta diversity, but when it's compared to the OTUs, they have a highest beta diversity. In case of the OTUs, when we consider the OTUs, they has the highest beta diversity. And the variation between the visits of the same person is less as compared to the between the subjects. this is both for the organism's composition as well as in the metabolic functions. And this will consider that this microbiota is stable over the time, and this is the unique feature of this human microbiome project. Similarly, when they analyze their data across the body sites have a no universal marker taxa. They are all different in the composition, but in case of the metabolic functions, they have a same constant metabolic activity they have represent in that community. In vagina only, little. No, they say that this is the constant potential, constant metabolic processes. Now come to the taxa level. So each and every individual has a signature taxa group when they have analyzed the 16S data. So these taxa group are present. For example, in the less diverse taxa are also significantly personalized. For example, in case of the buccal mucosa, the major taxa is, in case of the oral cavity, the major taxa is the streptococcus in all the oral cavity samples, but that is in the followed by Haemophilus in the supergingival plaque. Sorry, in case of the tongue dorsum, that is the super Haemophilus, and in case of the supragingival plaque, that is the predator, is a signature taxa. And these data are also complement to the metagenomic data also. So these signature groups are complement to the metagenomic data. So, and they have also analyzed that whether this healthy human microbiome contains any pathogenic organisms or not. So they have first checked that whether the NIAID reference NIAID class A to C pathogens are present or not, like Helicobacter pylori or Vibrio cholerae. So they are not present, but Helicobacter pylori they have found in only 2 stool samples. But they have also found the opportunistic pathogens based on this Patrick database. So these opportunistic pathogens are present in the ubiquitous form, near about ubiquitous form. They have also associated their microbial community with a phenotype. So in case of this vaginal pH has observed that correlate with the microbiome community. As the vaginal pH is increased, the lactobacillus species is decreased and the actinobacteria species is increased. Similarly, in case of the phosphate transport system are also increased as the as this pH is increased in the vagina. So the people are saying that if you in some body sites, if your microbiome diversity is less, then it is good. But in case of the gut, if your microbiome diversity is high, then it is good for a healthy person. So less microbiome diversity in the guts related to the obesity and the IBD. So these results suggest that the careful examination of the specialized metabolic functions will be key to deciphering the signature characteristics of each microbiome of the body, and we'll still know little about the most of these predicted genes and the proteins in the human microbiome. So many of them are not assigned to the any function. So that the next key step is to characterize the functional properties of the microbiome at both the strain and the total community levels. So the interesting question is what the most important factors are influencing lifelong microbiome composition, whether they are genetics, diet, birth environment, geography, or combinations of these factors. So these are the special collections from the Nature and the PLOS for this HMP data and its published articles. And they have over more than 200 publications from this project. And this is the best place to explore the HMP data, tools and the protocols. This is the hmpdoc.org website. So whatever I have presented over here, that is already in this website available. So different data sets are available, 16S as well as metagenomic data. Reference genome sequences are also available, that is sequenced by this group, and the tools and protocols are also available. So the next, that is the HMB phase 2, they have already launched it, and in that they have a multi-omics approach. So previously they are only based on the DNA, that is the metagenomic approach only. So now in the next phase they use the metagenomics, metatranscriptomics, metaproteomics, and the metabolomics. So this approach they are using in the phase 2. So thank you.
Tejas, in the first part of the presentation, when you were talking about sites for selection for samples, you mentioned heart in that. You mentioned heart.
No, I have not mentioned.
You mentioned it.
It was there in the presentation.
It just goes to maybe I may be incorrect.
But there
The
are
first part.
publications coming out now.
And that's why I want to understand actually. Hold on to that. Okay, just this. पीछे जाओ.
Yeah, here. Yeah, they have isolated some bacteria from the heart, brain, bone, spinal cord.
That's right. Okay. So these individuals were healthy, right?
Yeah.
Right? And they were having organism in their heart, correctly?
But the question may arise that how they have isolated from the heart.
You cannot question. You are yourself saying that they were having that much number of experts and all those things, and sampling error and all those errors. These errors have to be neutralized somewhere, right? So you are having microorganisms from brain, correct? You are having microorganisms from gut, correct? And still the subjects were healthy.
But they are not only healthy. They have a 15 demonstration projects are also that are disease related.
Yeah.
So it may be chances that they have isolated that organism from the demonstration projects. The body sites that are disease.
See, what I am trying to understand, I am not saying whether you are saying right, correct or incorrect. See, in last slide you showed that maybe the gut microbiota contribute to malnutrition and all those things, right? So what I am trying to say that when yesterday we were discussing that maybe microorganisms are having organ-specific or tissue-specific growth. If your subjects are healthy and still you are finding microorganisms from the brain or from the heart and a subject is healthy. So it means they are doing something there, and second part is they are being spared by our immune system. So they are desired there. That's why they are there,
Yeah.
right?
From the blood they have isolated around.
In blood, that is different thing, because if somebody is having infection and all those things, then we can consider it in that way. But as I mentioned yesterday, that from the breast milk and all those things, so if they are present in brain, they might be having something to do there.
That is what they have come up in next project. That is health and disease. First was just to find out what is the microbiome of the specific organ. Now they are going into the phase where they want to identify what is in the disease condition, and they compare between health and healthy and disease condition.
That's correct, madam.
They have not only isolated the bacteria from the healthy individuals.
Exactly,
They
that's what I'm
have
saying.
used the
And they are planning the
demonstrations,
definition of healthy individuals, right? And the subjects were.
May I add something?
Yeah,
here?
yeah, please.
See, these are all the things which are coming up recently. So, I mean, many things we believe that these certain tissues were sterile. Using various methodologies, people have been successfully able to at least isolate microbial DNA. I would not say the live microbes, but microbial DNA from these tissues using metagenomic approaches. And now we are wondering what we believed earlier, whether these tissues are sterile. it's really true or not. And I mean, brain is one example, but see, earlier it was believed that human milk is sterile. Till very recently we believed human milk is sterile. Now we know that it has 700 species of bacteria.
Right.
Same holds true for placenta. Earlier there have been several reports where people have isolated microbes on placenta, and they believe that that's the route by which microorganisms are transmitted from mother to fetus. There have been some stray reports on amniotic fluids also, that amniotic fluid contains, it is not only in case of infections, but even a normal amniotic fluid. Earlier amniotic fluid infections were correlated with preterm deliveries, but now even under normal conditions people say that there are microbes associated with amniotic fluids. Now the studies are being extended to other tissues like blood and brain and certain other tissues. Like earlier also it was believed that the microbes in the lung are only associated with some lung infections, but now it appears that healthy lungs also carry their own some specific microbes. So probably most of the internal organs, there will be some organisms which are naturally associated, and we would come to know more and more about them and their role, whether they are there as a contamination or whether they are actually there and have some role there. That, I mean, only time would be able to tell us that.
That is what I was trying to say.
Yeah.
That simple dogma that if microorganisms are there in tissue and then there must be some infection. That is
No,
incorrect.
that is not true. That is definitely not true.
That's why I was trying to say that if, because generally we say that if there is translocation, okay, this is going to be pathogen or this is going to be killed by our immune system. And as his sampling procedure showed that in brain, that individual was healthy and still they were getting organisms. So it means those organisms are desired there. They survive in immune system.
Yeah, maybe. I mean, the time when you do more extensive studies and sampling and get a little clearer picture, we would know more clearly whether these are naturally associated and what is the role, or whether they are just passively there.
Sometimes what happens, the person, he or she may be in the carrier state also. Carrier state, that is the state in which you cannot, the person who will not show the symptoms of that particular disease, but the organisms are already present.
That I agree.
And especially the typical example of such type of infection, that is the typhoid. In typhoid, once you will get the Salmonella typhi, that organism it will harbor in the gallbladder. And due to that reason, the person frequently gets the typhoid infection, that is recurrence, relapse. At that stage, it requires the removal of gallbladder.
Okay, one more thing. You mentioned that in future approaches, they will be doing metagenomics studies, and they will be obviously will correlating with the phenotype, right? So in what terms? I couldn't get it because there are 100 subjects, right? So what is the criteria for phenotype? What do you consider as a phenotype?
They are trying to understand the biology of the host as well as the microbiota. They are associating this microbiota biology to the host biology.
Phenotype, because you mentioned that.
Yeah, phenotype is based on this. They are trying to use this whole genome sequencing data from the. They have already collected the blood samples from the healthy individuals, and they have harvested the lymphocytes from that, and they have already prepared the cell lines for the future studies. So how this microbiota or particular microbial organism is interact with these cells, host cell.
Thank you very much, Mr. Tejas. Now I request Mr. Rajesh Ranjan sir to please come and felicitate a token of love and appreciation to Mr. Tejas.